Neomi Habib. I'm really happy to be here. I had the opportunity the last 2 days to really meet so many interesting people. I got research ideas. I learned how to make vinegar. So I think I already gained something. And I'm definitely going to implement some of these ideas. So today I'll just start by acknowledging the people who actually do the work, which are our students. And the reason I come with a smile every day to the lab. And so I think this is our greatest resource at LSEC, is our students. And it's a lot of fun to work with. And at the end of the day, when I say we did something, it's usually them and not me. So start by acknowledging them. So today I want to talk about, we all know our brain is amazing, right? And that's why we're here. We love it. But in some cases, something goes wrong and things don't go as expected. So it could be memory loss and cognitive impairment in Alzheimer's disease, which is a major focus in my lab. It could be changes with aging that make it such a huge risk factor for so many diseases like Alzheimer's and Parkinson's and so on. It could be during development in autism spectrum disorders. It could be after injury or stroke, right? So in many cases, we don't only want to explore how our amazing brain works, but also what goes wrong, what are these mechanisms that make it break, and how can we repair it. And today the emphasis will be mainly on technologies that we have in the last decade that are really, I think, are transformative and give us so much hope in a way to repair. So generally, this is what we do in the lab all day. I see it as a detective work. We just keep gathering clues along the way in order to get to the cure. So we start by really understanding the problem, defining the disease and its symptoms, then trying to understand the basic triggers. What is initiating this disease process? Then continuing to drill much deeper into the mechanism. What is actually happening in the brain once this trigger starts that is really kind of making the disease to progress? And all of this is hopefully leading to a cure. So the better we understand the triggers, the better we understand the mechanisms, the better and more efficient cures we can find. So that's what we're doing, and we keep jumping along this line of where we're at in our research. And so that's kind of putting you a little bit in our mindset of researchers. And so we're starting with the triggers, thinking of generally across diseases, but this is actually true for Alzheimer's in particular, is a lot of time we have a combination of triggers, right? So a trigger to a disease could be the genetic basis, where we inherit our genes and some mutations that we have in our genes. It could be something environmental, like a bacteria or a virus that initiates the process of disease. It could definitely be influenced by our lifestyle, our diet, the physical activity we do and other things. And age on its own, I'm putting it here because age on its own, there are these accumulating damages that occur that we want to prevent, because what happens to our brain with age is actually a trigger for a lot of diseases. And of course, on the other side, we have injury, which is something kind of abrupt, but or stroke that creates a lot of abrupt change. So we can't talk about everything, so I thought for today, actually highlight one of these aspects, which is the genetics point, because of the tremendous advantage we've actually made in genetics in the last few years. And it actually goes both with the trigger and the mechanism, and I'll talk about both. So first, kind of a short intro to genetics. I know a lot of you have a lot of knowledge in all of these fields, but really to understand the talk, we have to be on the same page. So let's just define some basic mechanism. And I stole this slide from a D. So our DNA, the basic of genetics is of course DNA, right? It's the genomic material we inherit from our parents, which really, I think every time I think about it, it's mind-blowing that this genetic code, which is 3 billion base pair long, of like a book, that really encodes how can we generate life, how does every cell in our body can perform its task. It has all this information in this one code. So this code is actually based on these three, we call it letters, but they're molecules, right? A, T, C, and G, which is amazing. We think about our language. We have so many letters and words in it. But somehow in our DNA, from these four letters, we create this very, very rich language that includes words and sentences and really defines every aspect of our body. And where is this code hidden? So of course, it's in the sperm and the egg, when this initial single cell that is created, the zygote that creates the entire life. But actually it's stored in every single cell in our body, right? So every single cell in our body holds the entire information on how to create a new human being. And this is amazing, but not every cell needs to execute all of this code at once. So the way cells work is they actually choose pieces of this code, translate these alternative smaller molecules called RNA that encode tiny pieces of these codes, and these pieces of these codes are used to generate proteins. And proteins do everything in the cell. They build the cell, they're the enzymes or drive different reactions, and they're part of signaling and talking between cells, talking with the external world, and so on. Okay, so this is the basic of life. This is the central dogma of biology. And so we have the DNA, the code, we have the RNA, and the proteins that execute the code within every cell in our body. Right, so this is the basic disease. Now what happens when we have a mutation in our DNA? This means we'll have the mutation in the RNA, and eventually we'll have a faulty protein. So a faulty protein can generate disease. A faulty protein can mean some protein can be missing, some protein can do the wrong thing at the wrong place in the wrong time, and that creates disease. If we're thinking of Alzheimer's, for about 5% of Alzheimer cases is inherited. It's an early onset Alzheimer's where by inheriting a mutated gene, we actually have almost 100% of getting the disease. And here's an example for a gene that is, when we have a mutation in it, we actually can get Alzheimer's. This is the APP gene, where you can see it here. It's sitting on the membrane of the cell, and if you have the mutation, it actually leads to a wrong cutting of this protein and aggregates that are called beta amyloid plaques. And potentially some of you already heard of them. This is one of the major pathologies of Alzheimer's, and most of the targets for disease, for drugs that have been made so far, have been against this amyloid beta plaques. Okay? So this is one form of a mutation that can cause disease. In other cases, we'll continue with the Alzheimer case. Most of other cases, we actually can't know based on our genes if we'll get sick or not, because it's not only about our genes, it depends on the environment and our lifestyle that all integrates into the risk for Alzheimer's. But our genes, we do have difference between us. Some of us are born with a higher risk of getting Alzheimer's, and some of us could even be protected against Alzheimer's. And this is something we inherit. So for example, this major risk factor for Alzheimer's that unfortunately is quite prevalent is in this gene called ApoE, apolipoprotein E. It's related to cholesterol metabolism. And if you have the variant number 4, here you can see variant number 4 versus variant number 3. This is like a scheme of a protein and how it looks. So even by eye, you can see there's a real structural difference here. Okay, so this small one base pair of a mutation, one point in the DNA that is wrong, changes the structure of the protein, which can increase the risk for Alzheimer's by a lot. Okay? So, but now that we know that, now we have a target, right? Now we have a target, so now we can take one step forward. Okay? So, we can take one step forward towards the mechanism of the cure. Okay? So that's how we go. So let's go back to our topic of our talk, which are these technologies are now enabling us to go through this process of understanding the triggers, the cause, and the effect much faster. And so now we are all experts in genetics, so we can start. So the first The first really major breakthrough I want to talk about is in the field of genomics and DNA sequencing, followed by the advancement in AI that I won't talk about so much, but really when you have a technological breakthrough, it can't go on its own. And here AI, I'll discuss it later, is really a critical point in driving genetics. There are another major breakthrough that Iran will talk much more about, which is related to cell engineering, our ability to manipulate a cell and its destiny. And another major breakthrough, which is the genome engineering. So we not only can read the code, we can edit it. So how does it go together? How does all of these technologies actually go together in our ability to better understand disease and repair disease? So let's put it on kind of like a flow chart. It's going to be a bit messy. So we're starting with the DNA molecule itself. So as I said, the first breakthrough is our ability to read the code. Is the technology that enables us to efficiently really read this, we call it DNA sequencing, to really read this entire DNA code. This is our first breakthrough. The next one is understanding the code, right? It's not enough to have in a computer file with 3 billion base pairs. I need to understand what it means. I need to understand where does a protein start, where does it end, what does it do. And this is a result of a combination of a lot of bioinformatics, a lot of AI and machine learning, with a combination of a lot of experiments and integrated all the knowledge of the entire community coming together. Okay? So now that we know how to read the code, we know how to understand the code, we can actually do some engineering. So we can manipulate cells to change them to do what we want to do, maybe correct them to do the right thing. And we can also change, which is much more recently, we can efficiently edit the genome itself. We can change the genome itself and actually correct the problem from its beginning. Okay, so this is kind of how it goes together in the flow. And we'll go a little deeper into most of these, especially on the genome sequencing and genome editing, we'll go a little deeper into that. Okay, so I know so we're talking about our first breakthrough, which is DNA sequencing, and I really want to explain why it's such a big deal. Okay, so today we can read the DNA code. We can do it very efficiently and very fast. We can have really minimal errors in these machines that read the DNA. And we can do it for many sequences at once, and we can actually do much more than that. We can not only read the DNA sequence, we can read the RNA sequence, and I'll talk about both, and why it's important to read both. Okay? So let's put this on a timeline so it will be better explained why this is such a breakthrough. So actually, we can read DNA sequences for a long time. Already in 1977, we were able to read DNA sequencing using a technology called Sanger. Okay, so Sanger technology was really a huge breakthrough where you can take a DNA sequence and read it. Okay, and to put this on kind of a time scale, in the 1990s they started the Human Genome Project, the project that says we need to have a map of the entire human genome. How much time did it take them to complete this Human Genome Project? Until 2003, where we had the first draft of the genome. So this took 13 years to try and sequence the human genome and read it and have it assembled semi-correctly. Now it's a little improved. Why did it take 13 years? Because we were relying on this Sanger sequencing, which is very slow, very expensive, and it required a lot of AI and machine learning to actually put all of this information together. So shortly after the Human Genome Project was published, the second generation of sequencing was generated. This is called second generation sequencing. And a little bit after that, we have the third generation sequencing. So the revolution in genomics actually starts here, when these new technologies arise. And this is because of two really important factors. The first is the efficiency. So here you can see how the efficiency over the years. You can see how many sequence of DNA could they sequence in a given, let's see if it's a day, yeah, given day. And you can see, since the generation of second generation sequencing, there's a huge uprise, so we can sequence so much more. Today we can sequence more than 100 million times more than we could do in 1980. Okay? That's one thing. The other thing is the cost. Cost is also a huge limitation, and these experiments are still very expensive, but originally in the year 2000, where they sequenced the human genome, it cost $100 million to sequence one individual. Okay? This is a problem, right? If we want to understand disease, one individual is not enough, right? So today, the cost really started dropping rapidly after the generation of the second sequencing, some competition that's healthy that came in, and today we're almost approaching the scale where we have $1,000 for a genome, and they could actually take it much lower. So these are the two factors that came in, and just kind of a bit more about the technology behind it. So in the Sanger sequencing, we took a single DNA sequence, really only one, had to amplify it so we'll have enough of it so we could actually sequence it, and then run it through the sequencing machine to read the sequence. So it could be like 1,000 bases out of billions. What happens in the second generation sequencing? We drop all DNA sequences that we have together into a tiny chip. There's a special way of amplifying all of these DNA sequences at once, and this is one of the major advances they made. And then we use imaging to really read the different sequences of all of these DNA sequences in one time. Okay, so we're doing it all at once. And you could see in my timeline that there's also a third generation sequencing. So today, most of what we do, we still do it in the second generation. But I do want to say one word about the third generation sequencing, because I just think it's really, really cool. So in both of these approaches, they actually didn't, when they moved to the second generation sequencing, they didn't really think out of the box. They thought about efficiency. How can we move from one DNA to all DNA sequences? How can we amplify faster and better? How can we read the sequences much more efficiently? Right? In the third generation, they already are thinking out of the box. They said, why do we even need to, if we don't need to amplify and sequence, we could just take one DNA sequence and immediately read it. How can we do that? And a lot of time in biology, in molecular biology especially, when we innovate, what we do is we actually look at nature, and we try to find solutions that are already there. This is true for many, many reasons. Like fluorescent proteins came from some algae in the sea, and so on. Okay, so what they did here, they actually found, of course I forgot to mention, sorry, that if we scale up the data, now we can't understand it by ourselves anymore. We have to go through a pipeline of machine learning algorithms that we had to develop from scratch to really how to analyze this data. Okay, so sorry for the confusion. Now back to our third generation sequencing. So this is one of the third generation sequencers that were developed. Notice what is happening here. This is a protein. It's like a channel that the DNA sequence can go through. Okay, I just think it's mind-blowing. And you can see the DNA sequence there just going through the channel. Okay? As it goes through the channel, there are slightly tiny moves depending if an A, C, G, or a T is passing by. Okay? And what the machine can do, it can read these small conformation changes in the protein, and based on that say what is the sequence. And it doesn't matter if it's a DNA or an RNA, if there's a modification, if there's a change, it can actually read that. So that's why it's really amazing. It could do for really, really long sequences. So it's great. I don't know why we're not using it more, because it, of course, has limitations. So it hasn't taken over the world yet, but it just shows this type of innovation that I find really fascinating. Okay, so what did we say so far? Basically, we said we have the ability to sequence. We have the ability to sequence many sequences. We can now sequence, I can today sequence a human genome, even a bunch of them, and get the results within a few days. This is a game changer, right? So this opened up the field of what we call the genomic era. It revolutionizes medicine, precision medicine, personalized medicine, evolution, plant biology, microbial biology. It's well beyond what our scope of our talk today about neuroscience. And let's go back though to the theme of our talk. Okay, so far we talked about this line where we want to find the trigger and the mechanism and the cure. So let's see how genomics integrates into this space of finding the trigger. Okay? So we said one of the triggers for disease is mutations, right? But we need to somehow find them. How do we find these special mutations that are disease-causing? So once we have the ability to sequence hundreds of thousands of individuals, we can actually do that. So we're taking a disease group and a control group, extracting DNA from everyone, measuring their DNA, and then using data analysis and statistics, finding these specific places in the genome that are associated with disease. Okay? I'll give you an example why the numbers are so critical. So I think one of the best examples I like, which just came out really at the start of where second-generation sequencing just started, is from schizophrenia. So schizophrenia is a disease that we know has a very strong inherited component. It was extremely hard to find any mutations related to the disease. And so, for example, this is a graph that shows different all the sequences of the DNA, and a location that is high means this is a place that is a specific region of the DNA that's associated with disease. And you can see here on the left that until 2011 there was just barely one single mutation that was linked to schizophrenia and could not explain the whole forms of the disease. In 2014, at the beginning, when just the second generation sequences arrived, they arrived at the Broad Institute where I was a postdoc, and they decided to do a huge world effort to solve this problem. They assembled more than 500 investigators from over 80 research institutions around the world in 25 countries, and they all gathered DNA from patients and controls, sent their sequences together, and assembled it into one lab who done all the statistics, and this really found this abundance now of DNA mutations that it can link to schizophrenia. So you can see here, because of the diversity, because individuals are so different from each other, it was very hard to find these driving mutations without having enough individuals sequenced. Okay, so today we're actually thinking of much larger data sets because the sequencing is cheaper. We can do it much faster. So we can actually look at diseases that are even more complex than schizophrenia. Okay. So this is one example. So we talked about how we can now use genomics and these advances of technology to find the triggers. Okay, so we found the mutation, and now what? Now we need to go one step forward into the mechanism, right? So how can we drill into the mechanism? We know where to look. We have a protein, but what do we do next? So we have to go back to our blueprint of biology, right, and understand what's happening. So when we want to look at mechanism, we need to see how the code is executed, right? So we said when we're looking at every cell in our body executes the code, which means translates RNA and proteins to generate the function. Okay, so if we want to understand the mechanism, we need to see what is wrong in the execution of the code. Okay, we want to compare a healthy brain to a diseased brain and see what molecules they're generating that are different, and hopefully find this kind of magic molecule that really is the root of this problem, that if we eliminate it or enhance it, we can actually reverse and find the cure. Okay? So how can we do that? We can do something very similar to what we've done before, only instead of measuring DNA, we're going to measure RNA this time. Okay, so we can take a group of disease and controls, measure their RNA, and then find these molecules that are changing, which tells us where the code is not executed correctly. So this is great, and it's actually the first RNA sequencer started when I was during my PhD, and I remember we're all sitting around and saying, like, how can we even analyze this type of data? What kind of noise is there? How do we even start and handle this? But today, this is something that every lab actually can easily do RNA sequencing. It's not even a problem, and we can better get a lot of clues on what is wrong in the execution of the code. But there is one limitation to RNA sequencing, especially when you're thinking of the brain. So this is like a cartoon snapshot that one of my students drew of the brain, which of course does not look like an actual brain, but it gives the message, where a brain has multiple cell types all combined together, right? So we can see here the neurons with the oligodendrocytes and the myelin. We have astrocytes, we have microglia, or the immune cells of the brain. We have the vascular system with the endothelial cells and the pericytes. And they're all together are very critical for the function of the brain. So what happens when we're doing RNA sequencing, we're actually taking all of this information together without the ability to separate it to the different cell types. So instead, during my postdoc, I've been developing this method that enables us to really measure these RNA, but separate it to the different cells. So what we're measuring is we're measuring RNA within a specific given cell types, and we're doing it, a given cell, not a cell type, and we're doing it for hundreds of thousands of cells at the same time. Okay. So this really gives us the resolution to go much deeper into the mechanism. Why is that? If we find a molecule that is faulty in bulk RNA sequencing, we took one step towards the mechanism, right? But if I can now tell you that this is done by oligodendrocytes, I took another very important step towards understanding better the mechanism. Okay? And that's so today also actually using so this is, let me just show you. The basic of this technology, which is now changing again actually, is these small microfluidic devices where we're flowing cells into a microfluidic device. It's encapsulated in small droplets that separate cells from each other, and then we can tag the RNA from each of these individual cells and look at it separately. Okay? So let me give you one example from our own research in the lab. So what we're trying to look at, just one second. Okay, what we're trying to understand is, as I told you, is Alzheimer's disease, right? And we had maybe before that, one second. Missing one slide. Start here. Sorry. Okay. So, you see? Yeah. Okay. So in Alzheimer's disease, we know there's a process moving from the healthy brain to the diseased brain, right? And this process takes over 20 years, 25 years. Okay, so just the last few years of the disease, we have the clinical symptoms we're identifying. And really our major quest was to understand what is happening throughout this process of the disease in the brain. How is the brain changing throughout this process, and specifically, what is happening during these earlier stages of the disease before any signs of clinical symptoms? Because this is the real therapeutic time window where we want to treat individuals. We don't want to treat them at the last stage as we are doing now. We want to transition to prevention medicine that will be personalized and tailored to the complexity of each individual based on their genome, based on their other comorbidities. If they have diabetes, if they have high blood pressure, the prevention therapy should be tailored specifically to them. So this is our vision that we're trying to walk really fastly towards this. But we first need to actually have this map of changes in the brain. Without that, we don't have our first clue on where to start from. Okay? And there's another really major question here, is because if we look at the aging population, we're also different, right? Not all of us are walking on the path of Alzheimer's. Some of us are working on some path of healthy aging, alternative brain aging, and in best case are the super agers, really age so well, right? And they can live for many years. What we want to do is we want to shift everyone towards the super agers and actually learn from them these mechanisms. So that's generally our our goal. And in order to do that, we decided to use our single nucleus RNA sequencing technology to profile these cellular maps of the brain. And this is a huge collaboration with researchers from Columbia University and Rush University in Harvard, where we took almost 500 human brains, and we profiled them with this microfluidic device that we have in order to generate a map of cells and how they're changing throughout disease. And we have almost 1.7 million cells. So this is a picture of how our maps look like. It looks like continents, but actually there's built of many tiny dots, and this gets 1.7 million dots, where every dot is a single cell. And we annotate them. We know which cell type they are, what is their state, what is their function, because what we're measuring is we're measuring RNA. So we know for every cell, how is it executing the code. Okay? So this is what we found. We've done a lot of analysis. Overall, it took almost 3 years to complete this study, and I'll now just tell you the bottom line. Okay? So the bottom line is that when moving from a healthy brain to Alzheimer's dementia, our first discovery was that if we look at all this complex of cells and we ask which cell is changing from the healthy to the Alzheimer's, what we discovered is it's not a single cell type that is changing. It's actually the entire cellular environment of the brain that is transitioning to this disease from a healthy state to a disease state. Meaning that we can't look at the cell separately, we need to look at this entire cellular environment as one unit where cells are communicating with each other, and when one cell is changing, the other one is following it. And some of these changes could be beneficial. It could be the natural protective mechanism of the brain. But a lot of this, at the late stage, is already harmful on its own. So a lot of the therapies in Alzheimer's are targeting these amyloid plaque proteins and trying to remove them. What we're saying is this is not enough. You have to also repair the cellular environment of the brain in order to restore this natural ability of the cell to protect itself and regenerate. So this is one conclusion, which is very important. But another, I'm taking us back to our questions. What we want to really know is we wanted to find all the dynamics of this process, and especially these early stages of disease, and really separate between aging and Alzheimer's. So I can't really explain the whole process, but using this new algorithm we developed, we actually managed to do that. We took all the 500 individuals and mapped them into a map of 2 distinct cellular paths. And we could show that some individuals are actually on the path for healthy aging and others are on the path of Alzheimer's. What is the meaning of this? It means that if we have a brain of an individual that has some pathologies in its brain, we can now, based on looking at the RNA in his brain, we can say if he's on the path towards Alzheimer's or he's on a path towards healthy aging, and either way, both of them, we want to have a cure to prevent further damage, but it's a completely different cure because the cells in their brain are doing something, are executing the code completely differently. Okay? So let me put this in a much more friendly way. This is how we look at it now. Okay? So we're starting from a healthy brain. A small proportion of us actually stay in this completely healthy state. So their brains, when they're 85-year-old, looks like a 50-year-old's brain. Okay? But a lot of us do actually transition with time, and that's okay. It's okay to be slower. It's okay to process slower. We actually had this discussion where we have many other things we gain with age, like wisdom and experience, right? But we want to prevent the path towards Alzheimer's, where we get the memory loss and dementia. Right? And we also want to slow down the path towards aging as much as we can. So we now understand there are really 2 distinct paths, 2 distinct ways the cells execute the code in a wrong way. And it starts at the beginning of the disease. It's not something that happens late. That means we have to target our therapies at these early stages of the disease where we're going on these 2 paths.
So now we can take some cells from my brain and tell me
I could if you so this is a really good question, Idan, because We can't, right? I mean, we can if you choose to, right? But no one would give
What was the question?
Oh, sorry. So we then asked, can I take some cells in his brain and tell him where he's at? So the answer is, if you give me a piece of your brain, and this is specifically the prefrontal cortex, I could. Actually, we've done that with simulations in our computer. We can take new data and project where they are. The problem is, I don't want you to give me a piece of your brain. That's very invasive. That's really harmful, right?
How big should you?
Yeah. You're getting it? Okay, great. I mean, I don't want you to open your skull, that's to begin with. So we need to translate that into a test we can do. Okay, so currently, we do have a new blood test, and we're running many individuals through that. But if we're looking at this path between aging and disease, our blood test detects individual here. So they're healthy, they're cognitively healthy, and we have a lot of hope for this new blood test and this new cohort of individuals that will actually be able to understand how to prevent their transition to the clinical phase. But it's not a complete solution because I think we should be able to detect people here. So we have now, but we took one step towards the mechanism, and now we need to go one step towards the cure, and the thing tying that are biomarkers. So we need a blood biomarker from our maps to find them in the blood efficiently. This takes a few years. We have candidates. We need to do the blood test, monitor individuals. Whenever we go into the clinic, everything slows down. Okay? That makes sense.
So sorry, I didn't understand very well.
Yeah.
The blood test is available or not available?
The blood test? Yeah, I didn't talk about that really in an ordered way. The blood test measures one of the major pathologies of disease called tangles or phosphorylated tau protein. It's a later stage compared to the amyloid plaques. It's another great drug target, and it's probably available in the U.S. We do it in Israel as a clinical study. We brought in the first machine. It's a collaboration with Hadassah, the neurologist in Hadassah. And so we've already done scanned a few hundreds of individuals to find individuals who are at this preclinical stage of disease. There is a question, do I want to take this blood test and know 6 years before I have memory loss and Alzheimer's to know if I'm on the path to Alzheimer's? Yeah. So anyhow, and you could only do it at a later age because the chances of having this age of, for example, 60 is completely irrelevant. Have to be, and 80 might be more relevant to try to find this diseases because it really captures the late stage of the disease, and this is a disease of advanced stage, right? So, this is the question. And I actually argue that it is good to know, but it really depends on your own personal mechanisms and how much stress is bad for you. But one of the reasons I think it is good to know, so in our clinical study we do provide some therapeutic intervention, but there's another aspect to Alzheimer's that we haven't discussed, so I'll just say it briefly. So we've been talking about the cells in our brain and how they're changing, but along the changes in the brain, we also have change in our aging body, right? And more, and I think more we understand in the Alzheimer's and aging field that we cannot disconnect. We can't look at the aging brain without looking at the aging body. Specifically in my lab, we focus on the immune system and in metabolism, because both of these are known to be influencing directly the brain. And I'll just give you one kind of little highlight from a new study we just came out, which is completely done in mouse models. Also what we do is in human brains, but this was done in mouse models in a collaboration with the Weizmann Institute, where we altered the diet of the mice and added more fat and calories, and this immediately, of course, affected the metabolites and the metabolism of these mice. And we were able to find a metabolite that induced an accelerated aging of the immune system. The immune system that's supposed to protect us against microbes and any insults is actually also regulating the brain directly. And what we've shown is that through the diet manipulates the metabolites that ages the immune system and accelerates the disease. So it's not causing the disease, it's accelerating the disease. Okay? So our hope is that we will be able to find, at least for now, at these earlier stages of disease, these type of systemic factors that we can use to manipulate them in order to slow down the progression of the disease or even prevent it. Okay? So this, of course, would go much better with your specific genetic background. So that's that vision.
What is the diet?
For the mice? So it's a bit complicated actually to translate that to the human, and I think it's one of our problems. Because if you look at the research, it depends on what is healthy diet, there's a lot of controversy there regarding like red meat, yes, no, ketones. But there are some things that are true. And to be honest, I always say to people, and I think all of you, I don't need to say that to you, but at least in Israel, I feel people in the US, of course, where I was there in my postdoc, people are not aware enough on how much control we have over our body. And the way our aging body, and the way I think about it is, let's say I tell you today, there is a magic medicine, doesn't cost anything, you can start taking it today, and it's better to start when you're 30, but you can start at any time, and it would not promise you won't have Alzheimer's, but it could affect 50% of your chances to get the disease.
Stop the sugar?
Yeah.
Stop the sugar?
Yeah. No, it's much more general. It's generally looking at just 1 second. See, it's going already one way into the cure. So it's this mantra of healthy aging. It actually is backed up with a lot of research, surprisingly. So we start with a healthy diet. So as I said, there's a lot of controversy regarding diet, but definitely we know that vegetables are good. We know that reducing carbs are good. We know that some type of energy balance at an early age is good, but not at the late age and not for children, for example. So it becomes a little complicated, but generally, if we think of what we eat, it's good, less sugar, of course. But it's not only that, it's related to our sleep, and we tend to overlook because we're working and we're stressed. It depends on our stress, which is so easy for young mothers with kids, right, to not be stressed. It depends on our oral hygiene. It depends on staying active cognitively and socially. And of course, a lot of work depends on physical activity. Okay, so it sounds like kind of a healthy aging mantra, but actually, there's so much research backing this up that we can't ignore it, and we should preach it to the general public, so from you to all your communities. So that's one aspect of the cure. How am I with time? I have no clue when I started. Oh, sorry.
So good.
Yeah.
15 to 20.
15 minutes?
I'm very intrigued by how stress, shocks to the system, activate the predisposition we have to activating a disease. Okay, and I wonder how much tests have been done. I have a neurological disease, and I know that it was a shock to the system that activated.
Right.
I'm a living example of it, okay? So I wonder how much testing is done around this stress level, this activation of the shock. Because we're all born, we're predisposed to certain diseases or afflictions. But this study, even in mice, has it been done to show the acceleration?
So, to be honest, not enough. We recently were looking more about it. There's a lot of work on stress. There's a lot of work of Alzheimer's. Not enough work about the convergence of the two. But it's still enough to know the strong connection. There is a difference between early life stress that changes our resilience, and that changes a lot of things. It's our tendency to develop some psychiatric disorder, because generally we're less resilient, versus a late life stress, which is not always bad. Like if I have a deadline for work, that's not necessarily bad stress. It's this other type of, especially not for me, maybe my husband, because he's a different type of person, right? So it's, so we have to not, don't need to go, too crazy about it, but there is a lot of work showing specifically molecular mechanisms. So what you said is exactly one of the major things people are showing. That stress modifies our immune system. And our immune system directly affects the brain. But there is more than that. When we're stressed, there are molecules that our body produces that directly regulate brain cells. So this is important because when we're stressed, we need to act in a different way. But if we do it chronically, then that's already bad, right? So it's this part. But generally, we want to generate this resilience. It's a cognitive resilience and emotional resilience, and that has been shown to be very helpful. Yeah.
Just a small question. Actually, we can take a lot of supplements. What do you think about that?
To be honest, I just don't know. It's like there's a lot of work about all kinds of supplements, and none of them, most of them, I mean, curcumine is very helpful, and that has been specifically shown. It reduces inflammation, and it actually seems to be working. But generally, there are a lot of supplements I'm sure could be great. There's just not enough good research to back it up yet. But definitely,
So we can change,
of course, yeah. Especially, so I'm not sure if adding just, if you have so what usually nutritionists say, I'm not an expert, but like what nutritionists say, if you have a healthy, balanced diet, you shouldn't be taking all these supplements because you have it from your diet. If you eat all the colors, all these kind of buzzwords. But if you're lacking something and you don't take the supplement, it would be bad. The best example is B12, right? You can actually damage your nerves, right? So it's obviously that we need to have or iron and so on, right? So okay. So moving on from this, I know this is like a hot topic because it's something we can actually do, and I had some amazing discussion and ideas on how to test diet in our new cohort that I'm going to think about from discussions here. But let's move on with do you remember our scheme? I'm jumping back to our scheme. So we started with our DNA, and we showed how we can sequence the genome to find the mutations. We can sequence the RNA to find mechanism. And both of these integrate into a lot of AI efforts, or machine learning as I call it, to understand the code. Okay? So what do we understand? We understand the DNA code, but we also understand the execution of the cells of the code. What do the cells do? What are they supposed to do? Okay? So this gives us enormous power to now step one more step towards the cure. But there are actually two more advances, technological advances, that make it much more feasible. So one of them is our ability, so now we're coming from reading and decoding, we're becoming engineers. Now we want to manipulate the brain in order to repair it. So we can engineer the cells, we can engineer them to manipulate them to do what we want, and Eran will actually talk much more about that. And we can engineer the genome and edit it so it can repair it this way. So I'm going to go through this quickly. So I remind you, we're talking about executing the code correctly, meaning we want, basically at the end of the day, we want the right protein, the right place at the right time. Okay? So we know how to read the code, and we can track cells and know what they're doing. So now we have the questions, how can we manipulate the cells to follow our instructions? Right? We can tell them to do something in order to repair, or we can try to correct them so they'll kind of take them back to the right track. How can we manipulate cells? So there are many ways to manipulate cells. Usually when we're thinking of drug targets, we're thinking of a small molecule. Let's see if this is going to go again. Yeah. There's like a small molecule that actually can connect to proteins on the surface of cells and activate them in a certain way. Okay? So here you can see like a surface of the cell, this line, and there's this tiny protein that could be a drug that we're taking that connected to a protein in the cell and generated a sequence of events. Okay? Go through it again. Here, follow this guy. Bloop. See? Join? Okay? So this is, we're generating a cascade of proteins that know what to do, but we gave it the first clue by giving it a small molecule drug. So we can find these, if we know what protein is wrong, what process is wrong, we can try to find this small molecule. It looks like it, but it's not. This is like, it's not a great picture. It's like a receptor sitting on the membrane of the cell. We gave it a tiny molecule that then started a sequence of events. It could also be a molecule that goes inside the cells. There are all kinds of ways, but generally it would be a small molecule that would bind a protein and generate a reaction. And this reaction would cause this cell to do what we want. Or it could activate a chain of reactions and crosstalk in the brain. For example, if I'm taking a neurotransmitter, it would activate a specific circuit in the brain, right? Okay. Another way we can manipulate the cell is actually give it a little piece of code. We know what is the code we want it to do. Why not synthesize it synthetically and deliver it into the cell? So we can deliver a small piece of DNA or a small piece of RNA. This might sound familiar to you. When did we all recently take a small injection of RNA that went into our cells and made the cells generate a protein that we wanted it to generate? Does it sound familiar? It's the COVID vaccine, right? The COVID vaccine is a capsule, is a lipid capsule that encapsulated an RNA that was delivered into our muscle cells, told the cells to generate the COVID protein so our immune system would identify it and will be vaccinated. Okay? So we can do that, and people have been doing it for years. Actually, before the genomic revolution, even just at the beginning of that, delivering DNA and RNA, it's not an easy task to do in the brain at all. It's one of our major challenges. Okay? And it could be a little scary, right, for delivering a DNA.
So you're saying trillion cells.
Right.
How do we deliver that? And each cell we have the DNA. So we have to deliver that to a trillion cells.
That's a really good question, actually. It's a major... So, this is... You're really raising some of the points. What is this? Why is this so hard? So, it says we have trillions of cells. They all have... Can you hear me? I'm repeating it.
Okay, so my question, you said that there are trillions of cells, that the DNA is in every cell. If we want to modify the DNA, do we have to modify the DNA in every of the trillion cells?
Yeah, that's a great question. So I didn't say modify the DNA yet. I'm going to say it in a second, but I'm going to answer the question because it's exactly the point. It's part of what we call, for example, if I'm delivering this RNA or DNA molecule, I was, for example, in the COVID vaccine, we delivered it directly to the muscle. Most of the cells affected were only in the muscle of the hand, right? So we did it locally. And it really depends on how do we define the problem. Surprisingly, the same mutation in the DNA doesn't always affect all the body. So a lot of time we're identifying the organ that needs to be repaired, and we try to deliver the cure to that organ. The problem is it's not so easy to have a specific therapy delivered to a specific region. A lot of times, for example, I think the best example is actually chemotherapy. We know exactly where the cancer is. A lot of times we want to target cancer cells, but we have a drug that we're taking orally that affects every dividing cell in our body, including our hair, right? So the problem is actually specificity and not the generality. So surprisingly, but it's a surprise, and we keep asking that question.
How it works?
What?
Copy. You give it to the muscle.
Yeah.
You put it in the lungs,
No, so what you do, that's a great question. No, so in the COVID is different. COVID, what we wanted the cell to generate a protein. We wanted our immune system to identify the protein. It didn't matter where it was. And that's why if later we have a virus in our lungs, the immune system could attack the lungs as well. But in other cases where we have a mutation in all our body, we might want to repair it everywhere. Right? But surprisingly, even though the mutation is everywhere, a lot of time a specific organ is attacked. It's a mystery sometimes. We don't understand it. The protein is expressed everywhere. The disease is only in the brain. We don't understand it. Maybe Iran will talk about it, maybe not. Yeah. So our next level is exactly what we were talking about. Not delivering a protein we synthesize, but rather correcting the brain. So this could be also for enhancement, but currently we're mainly talking about correcting mutations. 2 minutes. Yeah. I'll talk about what I have time for. No, okay, we're correcting mutations. So, major breakthrough. Just got a Nobel Prize for the CRISPR-Cas9, which is one of the machineries we can use to correct mutations in the DNA. Two amazing researchers, Emmanuelle Charpentier and Jennifer Doudna, just got the Nobel Prize. I think it's the first time that two women got the prize without any other men. I did put a man here. I'll refer to him in a second. But Emmanuelle Charpentier has been studying bacteria for years. That's her research goal. She's been studying the immune system of bacteria. It happens to be this CRISPR system. So the CRISPR system is the immune system of bacteria. At one point, she discovered a mechanism that says, how does this immune system reach its target? It reaches its targets through a tiny RNA molecule that can attach the DNA at the exact place, and then this immune system comes and cuts the DNA. It's supposed to be a foreign DNA that's not supposed to be there. So what engineers like Jennifer Doudna, and this is my postdoc advisor, Fang Zhang, what they were doing, they were saying, how can we now take this system? It's very simple, because all we need is the protein in a tiny RNA molecule that would direct it to its target. How could we use it to now correct any DNA sequence we want, or extract any DNA sequence we want? So this is a model of the CRISPR system. The protein is in purple, the green is the DNA, and you see in orange the RNA. The RNA guides the protein to the right place so it can cut it in the right place. And the reason this is so transformative, and that they got the Nobel Prize in such a small time frame, is that before CRISPR, it was a very hard and lengthy experiment to do. And now we can actually do it very fast and very efficiently compared to other things. So really it revolutionized the way we do science, and potentially, hopefully, it revolutionized also the clinic and our way to repair the brain. So I'll end with how can we repair. I'll just say that Fang Zhang did not get the Nobel Prize with them, but I do think he should have gotten it, because actually what we showed in the lab is the first time that anybody actually edited the genome of a live cell was done actually in Fang Zhang's lab. When I just joined there, so I had the advantage of kind of being at the end of this project and seeing their ingenuity about it. And in the end of the day, the system that everybody uses is the system of Fang Zhang. So it's always a question in science, who is that real inventor? So I had to give him credit. Okay, so let's see. Can it be used in the clinic? Definitely. It's not taken over because it's complicated, but there are many clinical trials already done. The most advanced way there's been used is actually in correcting the blood, not the brain. Why is that? Because we can extract the blood, correct it, and put it back in. So this is a disease called sickle cell anemia, where the red blood cells are completely transformed. They extract these cells, correct them, transplant them into the bone marrow of the patient, and then he's generating the correct cells. And it's been a huge relief for the patients, and this is really at advanced stages. There are many clinical trials going on. One of them was I think one of the more neuronal advanced clinical trials that are going on are related to curing nerve cells, but in the eye, where they're injecting the CRISPR system directly to the eye. The eye is like part of the nerve system, but it's exposed, unlike our brain that is very segregated, and they're taking these special cells called photoreceptors and after the repair, they're completely correcting their function and their structure. Okay? I'll just say, because I'm running out of time, I'll say that there is some first transition that is resembling a cure for Alzheimer's. It's not Alzheimer's. It's a genetic disease that affects the exact same protein, that results in the exact same pathology we have in Alzheimer's, these amyloid fibrils. And in this case, there is a specific mutation. And it's not only in the brain, it's also in the body. And this has already gone through clinical trials where they use the same delivery method as in the COVID cure, use these lipid particles to deliver the CRISPR system, edit it, and the faulty protein completely disappears. So this is, I think it's a good step forward, and it's very encouraging. Why is it not taken over? Because it's complicated, because there are many, many risks. It could cut the wrong place and create cancer instead of cure, right? There's a huge issue of delivery. How do we deliver to the brain? How do we deliver to a specific organ, not to everywhere else? And a lot of ethical concerns. If we can edit the genome to repair disease, can we edit the genome to be smarter? Can we edit the genome to have beautiful babies? Right? It's a huge ethical consideration that we're thinking about it as a community. So just to end, I'll kind of connect my talk with Eran's talk and say that all of this, we're talking about how to manipulate cells, but today we actually have an amazing way to manipulate cells that Eran will share with you more, where we're actually telling the cell who he is and what is his identity. So an alternative to actually trying and manipulating cells in our body, we could also, when thinking of repair, we could take cells from a different source and transplant them into our body to induce repair. So I'll just finish by saying, like, one of such work we actually recently are completing in the lab, where we use a model of spinal cord injury, and we're transplanting cells that are extracted from the olfactory bulb. These are cells that are important for the regeneration of axons in the olfaction system, but you can actually extract them from a live person, from the same person even that is injured, and after growing them in a dish, we're actually injecting them back into the spinal cord of these mice, and it was amazing to see that neurons grow and regenerate after this treatment. The mouse restored, not completely, restored some of its movement. And by using our single-cell technology, we could actually show why. Why is this so helpful for the regeneration? And the reason is that we activated some stem cells that are sitting in the spinal cord. Usually they're sleeping, they're dormant. And by actually inducing these cells who are healthy, they activated these resident stem cells and induced this regeneration process. So this is kind of a completely different route, and it takes us to the world of stem cells that Eran will talk more about. So this is all for today, and opens a lot of possibilities. So we went through, very briefly, through all of these technologies, showing you how they come together to really understand the triggers, the mechanism, and the cure. Okay, and I'll end by saying thank you. Because
why was it so important to have the 3D structural molecular of protein? You know that DeepMind and the EMBL, they create the database. But why is it so important to create new proteins?
That's a great question, but so I won't come up with this. So there's another huge effort in AI to go, it's a one step beyond what I talked about today, but I did show you some images of proteins and their structure. So if I know a mutation in a protein, I understand there's a problem. But until I actually see the structure, it will be very hard for me to understand what is the problem. If I see if it's exposed or covered, and also to design small molecules that would target specifically this protein, really goes strongly into what we call structural biology. So it's a whole new field of effort. I'll just say that we always thank the brain donors, because it's amazing, and that science is really a group effort. We work with a lot of collaborators. Our funding sources and donors are really an important and integral part of what we do in the lab, and of course the lab itself. So I have to acknowledge, really think of everyone as one big effort to help us advance science. Everyone comes from their own way of contributing. Yeah.
I have a question. You know we have a copy, right? Of
yeah.
The mutations that both couples have mutation? Is it only one?
Yeah, I completely ignored that, but thank you for raising it. So there's a genetic expert here who highlighted that there are 2 copies for every gene in our genome, right? We inherited one from our mother and one from our father. And because our father's mother and father was not identical twins, we have many differences in these 2 copies. So a lot of times, mutation only occurs in one copy and not in the other. We're talking about it?
It's a very good introduction.
It's a good introduction, okay. So Edmond is going to talk about that, and it's one of the challenges of editing. Not every disease needs 2 copies in order to be disease. Depends on if it's recessive or dominant. So a lot of times it's enough for us to correct one of the copies. It really, really depends on the disease. The hardest disease to cure are actually the ones that you have to correct both copies, because it's just stochastically we have less success with that. Yeah.
The technologies you highlighted probably have a lot of outcomes in preventive medicine, yet it seems that the bulk of the funds in the health system today goes to curative medicine. Do you think that in the future we'll see a more balanced effort between curative and preventive medicine?
I mean, we're definitely promoting that in the fields of aging and Alzheimer's, but also, like if you think of Parkinson's, for example, like what Habib was talking about, because I think the entire community understands we have to go to prevention medicine. So currently, we're at the stage of early detection. So you might not see, we can't create a cure if we can't identify an individual early. So a lot of, I think in the last decade, we focused much more on these biomarkers for early detection. Today, for some of the, like what we're doing now with the Alzheimer's, we have some way to identify the early detection, so we can now gather individuals and start to really understand how to do that. But it kind of takes a little slower, but you're really on point with that.
I think one last question, then we'll take a break.
When you manipulate cells, is it possible to mutate the DNA or not?
Yeah, that's a great question. So it's exactly at the point. So I think Iran might show actually how he mutates the DNA, but this system, the CRISPR system, what it does actually, it cuts the DNA and then the cell repairs it in a way that it completely changes the DNA. So a lot of times, researchers in the lab, we don't always, we try to correct mutations when we try to cure, but in the lab, we a lot of time we put the mutation in order to study it.
Okay, so